Today we will study about the kingdom Animalia. You know the features of kingdom Animalia or the features of animals. They are multicellular, they are eukaryotes, they have a definite symmetry, a form or shape. Then they can move from place to place for their necessities like food, etc., or shelter, etc. Then they have multicellular or cellular grade, tissue grade, or organ grade organization. Because there are so many unicellular form, but most animals show any of the organ system, tissue level or cellular level organization. You know about sponges, they show cellular organization. Then in case of diploblastic animal, it shows tissue grade organization. The organ grade organization is shown by the triploblastic animals. Those have ectoderm, endoderm, and mesoderm. Then there are heterotrophic, that means they obtain their food from plants. They cannot synthesize their own food. It depend on the plant for their food. Then they have a digestive tract, as well as they have they reproduce sexually by producing egg and sperm. Then they show an embryonic development, that means after fertilization, that means after the fusion of egg and sperm, zygote will form, and this zygote undergoes so many developmental process, and this zygote develop into a in case of direct development, the zygote itself develop into a miniature form of the organism. In case of indirect development, there are so many larvae. You already studied so many organisms or so many phyla like mollusk, arthropod, etc. They shows indirect development with the involvement of so many kinds of larvae are there. But in case of some other organism, for example, in case of us, we actually develop as a direct development without any involvement of any larval forms. So these are about the features of animals. Then next one is about the classification of the kingdom Animalia. You know that the kingdom Animalia is classified into three subkingdoms. First one is Mesozoa. Mesozoa include phylum Mesozoa and subkingdom Parazoa. Subkingdom Parazoa contain two phylum, Porifera and Placozoa. This figure is in your textbook, page number 40. And next one is subkingdom Eumetazoa. And this Eumetazoa is classified into two phylum or two classes or in two category based on their symmetry. Those who have radial symmetry, the animals with radial symmetry are categorized under radiata, and these involve phylum Cnidaria and Ctenophora. And the animal which possess bilateral symmetry, they are again categorized according to the some other features, that is protostomia and deuteriostomia, that we will see shortly. What is protostomia or what is deuteriostomia, etc. Then this protostomia and deuteriostomia, these are again classified according to their development of coelom, as well as coelom. Okay, these are platyhelminthes, nemaratia, they are nemertia, these are belong to coelomates, and pseudocoelomates are nematoda, nematomorpha, rotifera, etc. Then eucoelomate, or the also animal possess true coelom. They are annelid, mollusk, arthropoda. All these phylums are you studied in the paper. Then deuteriostomia, those involve ectoprocta, chordata, etc., that we will study in the second semester. Okay, chordata we will study, hemichordata, all this will you will study in the next semester. Then we have to discuss about the organizations and some other details of the animal kingdom. So first one, we will move on to the organization, cellular organization, body organization. First one, unicellular organism. You know some unicellular organism, for example, amoeba, paramecium, euglena. They have a single cell. Some organism they have cellular organization. These are unicellular organism. You studied about sponges. Sponges have a cellular grade organization. That means they have different group of cell. These cells are not organized into tissues. These cells actually work independently. They are not work as a coordinator manner. These cells perform independently, perform their functions independently. One more example is wallworks. In case of wall one more example is wallworks. I forgot to add the image of wallworks in this slide. Wallworks, they are actually a colonial organism. They have a group of cells, and these cells are not united into a tissue, or they are not organized into a tissue. But these cells are they are work to form, അല്ലെങ്കിൽ they can perform so many functions without the independently they can perform so many functions. Sometimes some cells are for reproduction and some cells are for nutrition. So there are so many cells are there, but these cells perform or എന്താണ് regulate their functions independently, or they perform their functions independently. That's about the cellular level organization. But in some cases, these organisms, those organisms possess tissue level organization. They have a group of cells or a tissue. What is a tissue? Tissue is a group of cells that are similar in structure. Okay, a group of cells are there, and these cells have a similar structure, and also they have a similar function, and all together they form a specific activity. A tissue is a group of cells that have similar structure and similar function, and all together they form a same function or a specific activity. For example, in case of Nidaria, we studied about the tissue level organization. In case of Coelenterate, they possess tissue level organization. Some group of cell perform a same function. Okay, and some they are diploblastic also. That means during the developmental process, or you know that during fertilization zygote will form, and this zygote undergoes so many divisions. You can see in this image there are so 8 celled stage. That means after mitosis a single cell is formed into 8 cell, 16 cell, etc. During a specific time period, or after so many divisions, there are so many process are there. For example, blastulation, gastrulation, etc. First process is fertilization. So sperm and egg will fuse together. This process is called fertilization. Then after fertilization a zygote will form. I will show you the image of zygote. Okay, I will draw some. So sperm is here and egg is here, so they will unite to form a zygote, and this zygote will undergo so many divisions. So they form 8 celled stage, 16 celled stage. Then there will be a mass of cell will form. This is called blastula. Blastula possess so many cells. Their scattered cells are there in case of blastula, and this blastula will organize. You can see in this image the blastula. This is blastula. After 8 cell stage there is another stage here, that stage is called blastulation, and that mass of hollow cell is called blastula, and this blastula undergo gastrulation and form gastrula. In case of gastrula, the cells are organized into ectoderm, outer layer, that is ectoderm, then inner layer, that is endoderm, then a middle layer that is called mesoderm, and there will be a cavity inside that is called archendron. Uh, so after fertilization, so many divisions occur. As a result, there are so many developmental stages occur. One of the stage is called blastulation and another stage is called gastrulation. During the time of gastrulation, the cells are organized into ectoderm, endoderm and mesoderm in case of triploblastic animal. The ectoderm will form our skin, nails, hairs, etc. Mesoderm will form our internal organs, for example, our alveoli, etc. So there are so many specifications are there. So in case of diploblastic animal, the organism they have endoderm and ectoderm. There will be no mesoderm. So the organism which shows tissue level organization, they are diploblastic. That means they have only ectoderm and endoderm. But there will be a mesoglia, that is an inner layer in between the ectoderm and endoderm. That is about the tissue level organization. Then organ level organization, that means they have the animals which shows organ level organization, they have a group of tissues are there, and these tissues are organized into organs for increasing the efficiency of that organ. So there are several tissues are there, and these tissues are associated to form organs to increase the efficiency of body function. Okay, for example, platyhelminthes and other more evolved group possess an organ level organization. There are so many organs or several organs that perform related function. Okay, in some other cases, these organs are again grouped into organ system. So these are called organ system level organization. This is about the cellular level, tissue level, organ level and organ system level organization. Next we will move on to the symmetry. Those organisms have radial symmetry is categorized under division radiata, and which possess bilateral symmetry, they are categorized under bilateral division bilateria. Okay, so what is radial symmetry? Radial symmetry, okay, first is radial symmetry. Radial symmetry means the in case of radial symmetry, their body, we can divide their body into two identical halves by cutting in any plane that is passing through the central axis of the body. So we can cut their body into equal half by cutting any plane passing through the central axis of their body. Okay, this is about the radial symmetry. Then next one is biradial symmetry. That means we can get equal halves only in two axes. We will get two equal halves by cutting the organism, or cut into two equal halves, but only when we dissect through two planes, but not more than two planes. So, in case of biradial symmetry, it's an intermediate step in the evolution of bilateral symmetry. That means this symmetry evolved before bilateral symmetry but after radial symmetry. You can see the symmetry in ctenophore. That means you will get two halves. But we have to cut the organism in when we cut the organism in two planes, we will get two halves, two equal halves. That's about the biradial symmetry. Then next one is bilateral symmetry. Bilateral symmetry means we will get two equal halves when we cut the organism through their sagittal plane. That means the mid region or their longitudinal plane, we will get two equal halves. This is about the bilateral symmetry. You can see this kind of symmetry in planaria, arthropoda, etc. This is about their symmetry. Uh, next one. Next one, so radiata and bilateria. Radiata means those organism which possess radial symmetries belonging to division radiata, and those organism possess bilateral symmetry belonging to the division bilateria. So again there are some other classifications are there: protostomia and deuteriostomia. Protostomia means there are large group of animals. The difference is that during the development, embryonic development, I told you zygote will develop into gastrula eventually. After zygote will divide, or as a result of mitotic division, the zygote will divide and form 8-celled stage, then a blastula and a gastrula, and this is called gastrula. In gastrula, the cells are organized into ectoderm, endoderm, mesoderm, and there will be a cavity called archendron. You can see the cavity inside. This cavity is called archendron, and this archendron is open into the exterior of the gastrula by a pore, by an opening called blastopore. So simply the blastopore is a small opening from the gastrula or from the archendron of the gastrula. So this opening is called blastopore. So after the development, in case of proteostome, first one we will deals with the proteostome. So after the after gastrulation, there are so many events will occur, and this mass of cell will develop into a complete organism. The blastopore, in case of protostome, the blastopore will develop into mouth of the organism. If the blastopore will develop into the mouth of organism, it is called, this kind of organ or this kind of development is called protostomes. In case of deuteriostome, the archendron, you can see the same gastrula, cleavage is here. After 8-cell stage, there will be a blastula, then blastula undergo gastrulation and form a gastrula, and you can see the cavity archendron here, and the archendron open into the exterior by the same opening called blastopore. In case of this deuteriostomes, this blastopore will develop into anus of the organism. So this kind of development, or those animal possess this kind of development, is called deuteriostomes. This is about the protostomes and deuteriostomes. We can see spiral cleavage here. That means cleavage means the division of the mitosis or the division of the cell. That's only the cleavage. Spiral cleavage means the cleavage will like this only. Spiral cleavage, that means the division is like a spring like division. Okay, mitosis, there are so many kinds of division according to the organism: spiral cleavage, radial cleavage, bilateral cleavage. Okay, in case of nematode, flatworms, they will show spiral cleavage. That means the cleavage in this fashion, in spiral fashion. That is about the spiral cleavage. The protostomes show spiral cleavage. Deuteriostomes show radial cleavage. These are different types of cleavage that you will study in details of this cleavage in developmental biology classes. Uh, Here you want to study about the proteostomes and deuteriostomes. The proteostomes exhibit spiral cleavage and deuteriostomes exhibit radial cleavage. So in case of protostomes, the blastopore will develop into the mouth of the organism, and in case of deuteriostomes, the blastopore will develop into the anus of the organism. That's about the protostomes and deuteriostomes. Next we will move on to the coelom. Of the development of coelom, and what is coelomate? A coelomate, we know that coelom is the space between the gut and the body wall. You can see small space in between the gut as well. This is the digestive cavity or gut, and this is the body wall. You can see a space here. This is about the coelomates. These are coelomates. Annelid, mollusk, arthropods, echinoderms, chordates. These are coelomate animals. That means they exhibit a space between the gut, that is the digestive cavity, and their body wall. As well as this cavity is mesodermally derived. That means you know that there are three layers: ectoderm, endoderm, and mesoderm. And the ectoderm will develop into skin, epithelial tissues, etc. And mesoderm, each of this layer will develop into specific part of our body. This cavity or this lining of the wall, body wall, that is derived from the layer mesoderm of the gastrula. So that is eucoelomate. These are called eucoelomate organism. Then in this coelomate, there will be no cavity, no such cavity in between gut and body wall. This is about the coelomate. But in case of pseudocoelomate, there will be space is there. In pseudocoelomate, a fluid-filled cavity separates the digestive tract and outer body wall. The cavity is there, but there will be no lining by the mesoderm. There will be no lining of mesoderm in between the cavity and body wall. So these are the coelomates, eucoelomate, and pseudocoelomate. Coelomate, there will be no cavity, but in case of example is flatworms. But in case of eucoelomate, there will be a cavity there will be a cavity in between body wall and gut, and this cavity is the body wall is completely lined by mesodermally derived coelomic epithelium. But in case of the pseudocoelomate, there will be no mesodermal lining of the body wall. This is about the coelom development. This is about the coelom. Then next one, how this coelom is formed. Okay, in case of, you can see these are flat worms. Okay, a coelomate animal, there will be no cavity. But in case of the coelomate animal, that is annelid here, you can see a cavity here. Okay, coelom, this cavity here. But in case of a coelomate animal, no cavity. But in case of the pseudocoelomate, there will be no lining. Mesodermal lining is here. But in case of coelomate, there will be cavity as well as mesodermal lining. Okay, next one is about the formation of coelom. Okay, it's in your textbook, page number 46. Okay, first one we will discuss about the schizocelic development. Schizocelic development means you have the body wall here, then this is the endoderm, this is the mesoderm. Okay, mesoderm. So the mesoderm is formed as a split. We have you can see archendron here. This is archendron, and mesoderm will originate here as it is called a lateral plate mesoderm. Okay, so the band like mesoderm originated in two sides of the archendron is called a lateral plate mesoderm. It's a mesoderm only. Then this lateral plate mesoderm will split into lateral plate mesoderm are there, and this will split and develop as mesodermal band. This split will extend and form coelom. This development is called schizocelic development. So that means first there will be a split of coelom will develop, lateral plate mesoderm. This will expand and form the coelomic cavity in case of schizocelic coelom development. But The next type of development is called enterocoelic development. That means in case of deuteriostomes, the mesodermal epithelium that is originated as a split. That means outfold of the wall of archendron. This is archendron. So the mesoderm will originate as outfold of the wall of archendron. This kind of development is called enterocoelic development. So there are two kind of coelom formation. First one from the lateral plate mesoderm. You can see lateral plate mesoderm here, and this lateral plate mesoderm expand and form the coelomic cavity. This is the first type of development called എന്താണ് schizocelic development. But in case of enterocoelic development, one minute. In case of enterocoelic development, you can see that the archendron, the cavity of archendron will grow as an outfold, and this will develop into the coelom or the cavity between gut and body wall lined with mesoderm. This is called enterocoelic development. Then you already studied about the segmentation in arthropoda annelid. Segmentation means the segmentation is developed during the during the embryonic development. Okay, as a result of so many molecular changes, their externally this segmentation is exhibited by small constriction of their external epithelium or their external body part. Internally, it is exhibited by the internal arrangement of so many internal organs. Then, but some organism shows pseudo-segmentation. Pseudo-segmentation is exhibited by so many organisms. For example, tapeworms. Platyhelminthes exhibit pseudo-segmentation. That means the segmentation is not an actual segmentation. These are no true metamerism. Pseudo-segmentation is exhibited by platyhelminthes. One difference is that the true segmentation and the pseudo-segmentation, one difference is that in the position of zone of growth. That means in platyhelminthes, it exhibits pseudo-segmentation. The zone of growth is in the neck region. But in case of actual segmentation, in case of arthropod, annelid, etc., the new segment will formed from their posterior part of body. In case of pseudocoelom, new segments, that means proglottids in case of platyhelminthes formed from the anterior neck region. But in case of a true segmentation, new segments are formed from their posterior region of their body. That is the difference between pseudo segmentation and true metamerism. That's about the pseudo segmentation. So have to study about the segmentation, pseudo segmentation, symmetry, different types of symmetry, radial symmetry, biradial symmetry, and bilateral symmetry. Then what are different types of coelom, development of coelom, different types of coelom, as well as the classes of development, that is cellular level organization, tissue level organization, organ level, and organ system level organization, as well as the classification of kingdom Animalia. If you have any doubt regarding the classification or any type of development, you can message me. Okay, thank you.